P.2 G.1. I'm Jade and I'm 2.8.
My name is Mark Alonzo and I'm 2.2.
My name's Max and I'm 2.11.
And I'm Sarah Hitchley, 2.10.
Okay, so this is the cell cycle. In the cycle we have interphase and then we have the M phase. In the interphase we have G1, the S phase, G2, and then in the M phase we have mitosis and cytokinesis. So in the G1 phase, the cell grows, and so we make sure that the molecular building blocks are there, and the rest of the steps, and that the organelles are properly grown. And so then in this checkpoint here, we make sure that the cell makes sure that it needs to be divided, and if it doesn't, then it'll go into the G0 phase. And then this is a state where it'll remain, and it continues the cycle, but it doesn't need to be divided. And this occurs for cells such as neurons. And then in the S phase, the DNA is replicated, and then in the G2 phase, the cell makes sure that the organelles are ready for division in mitosis. So this is just prep for mitosis. And this checkpoint here, it makes sure that the daughter cells, or the cells, can be replicated properly. And then in the M phase, in the mitosis, this checkpoint here makes sure that the spindle fibers are there, so the cells will probably be able to divide. So in prophase, we have our chromatids, and then they begin to condense to form. The chromatin condenses to chromosomes, and then we have our centromeres, which are here, centrosomes, which are here, and then the spindle fibers. So the spindle fibers begin to form within the centromeres.
Centrosomes.
Sorry, the centrosomes. They begin to form between the centrosomes, and then the sister the chromosomes, they start to attach to the spindle fibers, but they first have to go to their polar sides. They begin to attach the spindle fibers, and then, but they're not aligned yet. No, I don't. Okay, well, I'm just gonna pretend that. Okay, and then it's not aligned yet, and then it has to go through the metaphase plate in order to align them.
Okay, well, in metaphase, the chromosomes line up.
And then the nucleus dissolves.
Oh,
The nuclear envelope
nucleus
dissolves.
In prophase. Now we're in metaphase. In metaphase, they begin to line up, and then just these rates. And this is where the checkpoint occurs. At the checkpoint, we check our regulators check that the spindle fibers are properly connected to the chromosomes, and we go to not telophase. We go to anaphase over here. Let me go over here. And this is where let me grab these. The centrosomes actually pull apart the sister chromatids into their own, now dividing cell. Are you good? Okay. And here, after they've been divided, that's anaphase, and now we go into telophase. In telophase, the spindle fibers begin to disappear. The centrosomes stay there, but they don't do anything until the cell divides again, when the daughter cells become fully developed actual cells. And then here, the nuclear envelope begins to reform. And the chromatids decondense into chromatin. So pretend this is like a soup now of chromatin. And then during cytokinesis, which overlaps with mitosis, in telophase,
So what he just did was telophase.
I said telophase.
Sorry.
And then now we're entering cytokinesis, which overlaps with mitosis. And then in cytokinesis, the two cells actually break up, and they become two daughter cells, which are genetically identical to the original. And I'm quickly going to talk about cyclin. Cyclin is a group of proteins that are regulators for the cell cycle. they regulate cell growth or they facilitate cell growth or hinder it. And cyclin bonds with an enzyme called cyclin-dependent kinase. Cyclin-dependent kinase is an enzyme that bonds with cyclin so that in areas in like checkpoints, it becomes more active, which it could help create cell death, or it facilitates cell growth. Thank you.
And now we're going to move on to meiosis 1 with me, X. And now I need to get And in meiosis 1, we start off with prophase, so prophase 1. In prophase 1, the chromatin condenses into, instead of two sister pairs of chromosomes, it forms into 3 homologs, which are two pairs of chromosomes put together into this. From here, crossing over occurs, which is basically the homologs exchanging bits and pieces of themselves in order to increase genetic diversity. This occurs to basically make new traits in a population. And lead to adaptations and new traits being introduced within the gametes. Once crossing over occurs, the centrosomes also begin to form their spindle fibers to prepare for anaphase. During this, also, the nuclear envelope dissolves. From here, we move on to metaphase. The centrosomes have moved to either pole of the cell, and the homologs then move to the middle of the cell, or the metaphase plate. They assemble at random, they still assemble in a line, but in a random order, in order to also try and increase diversity within the gametes. From here, we move on to anaphase. In anaphase, the centrosomes pull apart the homologs into their 2 chromosomal pairs. From here, we move on to telophase 1. In telophase 1, either cell gets 3 pairs of chromosomes, and the centrosomes begin to dissolve their spindle fibers. And from here, the nuclear envelope once again reforms in both cells, in both new daughter cells that will be formed. And the chromosomal pairs break down back into chromatin from here. And then cytokinesis occurs, cytoplasm then splits the cells into 2 new haploid daughter cells.
Okay, and then now these 2 daughter cells will each individually undergo meiosis 2. So we start with prophase 2, so the chromatin condenses again into chromosomes, and each chromatin matches with its sister to form 3 chromosomes in the cell. And then the centrosomes will appear with their spindle fibers. And then they will undergo metaphase 2, where the nuclear envelope will break down, and then the chromosomes will line up at the metaphase plate. The spindle fibers will attach to each sister chromatid. And then they will undergo anaphase, so each chromatid will be pulled from its sister, like so. And then they go into telophase, so over here. The spindles will disappear. as well as these centrosomes. Okay, and then in telophase,
Telophase.
in telophase 2, so the nuclear envelopes will reappear and each cell will contain 3 chromosomes or 3 separate sister chromatids. And then they'll undergo cytokinesis where the cytoplasm will split and then we will end up with 4 daughter haploid cells.
Thank you.